In section 9.4, we talk about the energy that's necessary for the muscle contraction, the mechanism of which we've discussed in previous sections. What we see is that there's no other there's no other cell type in which the rate of ATP breakdown is as variable as muscle fibers. And so the change is 20 to up to 100-fold from one moment to the next as a muscle goes from a relaxed to a contracting state. And so what this means is that because ATP is not a molecule that can be stored in large quantities, the muscle must have some mechanism to very rapidly generate ATP so that there's not a shortage of ATP as the muscle begins this contracting state of contraction. The amount of ATP that is present at the beginning of contraction is not sufficient to sustain contraction for any extended period of time. So there are three mechanisms that muscles use to generate ATP. One is creatine phosphate, one is oxidative phosphorylation, and the other, of course, is glycolysis. We see all of those illustrated in figure 9.22 from our text, shown here in the lower left-hand corner of our slide. And we go ahead to talk about these a little bit on this next slide. We begin with creatine phosphate. And so creatine phosphate is a way to regenerate ATP very quickly. In fact, what we notice is that in a resting muscle, the creatine phosphate concentration exceeds the ATP concentration by about 5-fold or so. And when muscle contraction begins, the creatine phosphate concentration decreases rapidly, but the ATP concentration remains relatively constant because that transfer of energy from creatine phosphate to ATP is seamless, and it happens so rapidly that it prevents the overall levels of ATP in the muscle cell from dropping. Now, creatine phosphate is not a long-term method of providing energy for the cell, and so creatine phosphate is only able to sustain contraction for a matter of seconds until either oxidative phosphorylation or glycolysis takes over, depending on how intensely the muscles are contracting. The last thing to say about creatine phosphate is that the amount of energy that's stored in that bond between creatine and phosphate is about the same as the energy that is found in that third phosphate and ATP. So when that bond is broken, the creatine phosphate bond, and that energy as well as the phosphate is transferred to ADP, then that allows us to regenerate ATP, and then that, of course, can be used by myosin for muscle contraction. Now, as we mentioned, that's only going to last for a couple seconds, and so we go down here to our next bullet point, and what we see is that after a couple of seconds, then either oxidative phosphorylation or glycolysis must take over. If the ATP usage is greater than 70% of the maximum rate of usage, then glycolysis is going to be the mechanism of choice for generating ATP. And of course, glycolysis, as you realize, is a good short-term method to generate ATP under anaerobic conditions. But of course, the byproducts of glycolysis, some of which we talk about in the next slide, are going to cause problems for the cell and prevent it from generating ATP via glycolysis for long periods of time. So for long periods of sustained contraction, the cell must rely on oxidative phosphorylation. And if we go up to this bullet point, sources of glucose for oxidative phosphorylation, what we see is that the energy for oxidative phosphorylation varies depending on how far we are into the exercising period. And that's assuming, of course, we're at less than 70% of maximum ATP usage. So for the first 5 to 10 minutes, we're using muscle glycogen. For the next 30 minutes, we're using blood glucose and fatty acids. And then after that, we're relying primarily on fatty acids to feed into oxidative phosphorylation and create ATP. Eventually, of course, muscles fatigue. And so what we see is in little figure on our slide here, which is figure 9.23, we see that we have an isometric contraction creating tetanus in the muscle. It can only sustain this contraction for so long, and then it becomes fatigued. And then we undergo a second contraction after a brief rest period, and fatigue shows up much more rapidly in that second period because of the energy exertion that we've already undergone in the first tetanic contraction there. And so muscles fatigue for different reasons. We basically divide these up into chronic and acute mechanisms for muscle fatigue. So in acute fatigue, what we see is that there are a couple different mechanisms that cause fatigue. And the fatigue is different than the fatigue that we observe in chronic muscle fatigue. So in acute fatigue, we see, for instance, a muscle that gets tired after perhaps a weightlifter picks up a huge weight, holds it above his or her head for as long as they can, and then drops it to the floor. So that's a good example of acute fatigue. We have muscle that's contracting rapidly, very suddenly, and then there's a quick recovery after that. And although the contraction that's possible may not be quite as intense as the original contraction, recovery does occur in a relatively rapid manner. Part of acute fatigue, interestingly, and maybe a little disturbingly, is the interruption of blood flow to the muscle. And as we will see in the cardiovascular system, your blood vessels, to some extent, are compressible within muscles. And so when the muscles contract, they may occlude blood flow, or they just make it a little bit more different. They're difficult, It may be more difficult for blood to make it through those blood vessels that are found within muscles. And so when the muscle relaxes, of course, blood flow is restored to that muscle. But we also have additional mechanisms, and so there are changes within the cell itself. So we use up ATP. In acute fatigue, we are generating ATP using glycolysis, and so we have very rapidly higher levels of ADP, higher levels of hydrogen ions, and changes in the muscle cell as a result of that, which lead to the following results, would make it more difficult for the muscle to contract. And so we divide in acute, again, this is all acute under this bullet point, we divide the effects of acute fatigue in the muscle cell up into these three categories. And so one result of this higher levels of ADP and higher levels of hydrogen ions in the cytoplasm is that we have less calcium released by the sarcoplasmic reticulum. And so, of course, less calcium released means you have less binding to troponin, less moving of tropomyosin, and less availability of the myosin binding heads on actin. We interestingly have less thin filament response to calcium. Even if we have calcium, the troponin molecules of the thin filaments are less able to respond to that calcium. Interestingly, eventually we have inhibition of power strokes of myosin. And so all these are observed in acute fatigue, eventually resulting in lessened ability of the muscle to contract. And so, again, hopefully to be clear, acute fatigue can be caused, we divided it up here, into two different mechanisms. One is the blockage of blood flow. The second would be the changes in the actual cell that are observed and the effects on the mechanism of muscle contraction that we list right here. Now, interestingly, chronic fatigue seems to be quite a bit different than the mechanism of chronic fatigue seems to be quite a bit different than the acute mechanism. And so what we see is that in chronic fatigue, one thing that we notice is that the ryanodine receptor channels in the sarcoplasmic reticulum that we discussed previously become leaky, and lots of calcium leaks out. This calcium activates proteases that actually work to degrade the muscle proteins that we've discussed previously, myosin and actin, and perhaps others. And so this leads to both a decreased ability to contract and soreness of the muscle that's eventually, of course, repaired during a resting period. But this is quite different, a completely different mechanism than what we observe in acute fatigue, as we discussed previously. Also, as would not be surprising to us, it does appear that depletion of fuel, not ATP, but depletion of fuel for oxidative phosphorylation is a factor in creating chronic fatigue within muscle cells.